Hey guys, good afternoon. We're going to start the chapter 10 today. And chapter 10 is sensory physiology. We will have two parts of chapter 10 today, part 1. And on Thursday, we're going to do part 2. So today we will talk all about general sensation. And on Thursday, we will talk about spatial sensation. So objective and outline for sensory physiology. We will talk about sensation. There are two kinds of sensation, general sensation and a spatial sensation. General sensation can be perceived by every part of your body, whereas spatial sensation can be perceived only by special organs in the body, and those organs particularly lies in our head region. Then we'll talk about categories of sensory receptors. Sensory receptors are several types based on what kind of stimulus they perceive and transduce. And then receptors produce receptor potential, means action potential produced by receptor. That is also called generator potential. We will talk about how your receptor produce the potential or action potential. Then we will focus on cutaneous sensation. Cutaneous sensation is the sensation in the skin. Guys, can you please mute everybody?
Professor,
Uh-huh.
I have a question. Last week we were talking about the brain, and then
Okay, can we, I will finish this lecture, and then if we have left, we will talk about those part today. I know there are certain part left, I think. I will go over that.
okay, thank you.
Okay, so I saw the message in your chat. I will work on it. Okay, so the cutaneous sensation means the sensation of your skin, and skin had several receptors like touch, pain, vibration, pressure. And then we will talk about how we determine which area of the skin are very sensitive. And to determine the sensitiveness of particular skin area, we measure receptive field and sensory acuity of the skin. And that is performed by 2-point touch threshold using the calipers. We will do that in the lab on Thursday too, okay? Then on Thursday, we will talk about a spatial senses. A spatial senses had taste and a smell, hearing and equilibrium, which is the function of our inner ear, cochlea, and vestibular apparatus. And then the vision, which is the function of your eyes. We're going to talk about these spatial senses in next class, okay? So sensation are two types. General sensation. General sensations are touch, pain, temperature, pressure, vibration, tingling, tickling. Those all are general sensation. Whereas spatial sensation are vision, hearing, balance, smell, and taste. The organ for vision is eye. For hearing and balance is your ear. Smell, nose, taste, tongue. And these all spatial sense organs are found in our head region, whereas general sensation are found everywhere in the body, okay? So the starting point for sensation is the receptor, whether it is exteroceptors, the receptors which are found in outside the body, like in skin, or inside the body, like osmoreceptor, pressure receptor, thirst receptor, hunger receptors. Those all are interoceptors. So receptors are interoceptors and exteroceptors. Let me write on that. Exteroceptors and interoceptors. So exteroceptors are the receptors which are found on the surface of your mucous membrane or your skin, like touch receptor in your skin. And interoceptors are inside the body. For example, like temperature receptor in our thalamus, or hunger receptor in our thalamus, osmoreceptors in our blood vessel wall or in our brain. Those are the interoceptors. Okay. So these receptors can detect the change in the environment and then produce action potential by depolarizing itself. Okay. And those receptors are called sensory receptors. So sensory receptors are dendritic nerve endings, specialized neurons, or specialized epithelial cells associated with sensory nerve endings. If you remember a skin diagram, you're going to see how these receptors are attached or holding on to the nerve endings. These receptors transduce, means they change environmental information into APs or action potential. And these action potential or electrical impulse is the common language of our nervous system. Okay. So there are several kind of receptors, and each receptor can change the stimulus of only one type. Not a single receptor can change several kind of stimulus. So each type respond to a particle form of information, which is called particular modality of information. For example, sound, light, and pressure. Sound receptor is perceived only by receptor in our inner ear. Light receptors, light is perceived only by receptor in our retina. Pressure receptors are perceived by the pressure receptor in the skin and another part of the body. Okay, so these different modalities perceives are different because CNS pathways they stimulate are different. So now question comes, let's imagine in our skin we have touch receptor, we have pressure receptor, and we have pinching pain receptor. Three receptors are in your hand. Three stimulus, three stimulus activated, and these three stimulus then create action potential, which is the common language, goes to your brain. But our brain perceives them touch as touch, pain as pain, pressure as pressure. Why is that? Because these nerve fibers with action potential goes to different region of the brain where they are perceived by association neurons. Are you following me? That's why different modalities of stimulus is perceived different in different part of the brain, even though they all carry only the impulse, electrical impulse. Okay. So let's see some example of the receptors here. You see here, this is the nerve endings and then some connective tissue attached to it, Merkel disc, Meissner's corpuscles, which is touch receptor. There is nerve ending, and on the nerve ending, you see connective tissue firing, clear? Then there is Ruffini's endings, again nerve fiber, and then connective tissue layer. There is Pacinian corpuscles here, So you have nerve ending going inside and then layers of connective tissue. It is like onion layers on the nerve fibers, and posterior corpuscles are for the pressure receptors. So these receptors can be simply a dendritic endings of nerves or a specialized ending of neurons or non-neuronal cells. In most of the case, they are epithelial cells. Characteristic of sensory receptors. Based on what kind of stimulus a receptor transduce, they are grouped in several functional categories. And the functional categories of receptors are chemoreceptors, which sense chemical stimuli, photoreceptor, which transduce light, thermo means temperature, thermoreceptor respond to temperature change, mechanoreceptors respond to deformation of their cell membrane. So when you press your skin, some of the epithelial cells plasma membranes are deformed, and that creates the action potential. Nociceptors responds to intense stimuli by signaling the pain. Okay, so nociceptors respond to pain or intense stimuli. Proprioceptors signal positional info of body parts and orientation. So when your body is moving one way or other way, when you're moving your hand, these receptors are activated and they transduce them. Okay. So classification of receptor based on their normal or adequate stimulus. They are mechanoreceptor, pain receptor, chemoreceptor, photoreceptors, and they are mechanism and examples are here. You can see them. So what does a receptor do? A receptor transduce external stimuli. That means it changes external stimuli. Sensory receptors also can be categorized according to location where they are. They are cutaneous receptor and spatial sense receptor. Cutaneous receptors are near an epithelial surface. That means either on your skin or the epithelial lining of your mucous membrane. These cutaneous receptors respond to touch, pressure, temperature, and pain, whereas spatial sense receptor are part of sensory organ. And those sensory organs are such as taste by tongue, hearing by your inner ear, sight or vision by your eye, and equilibrium or balance also by your inner ear. Okay. Okay. Before I go to sensory receptor responses, let me give you one example. So when you buy a new perfume and you spray on yourself, its fragrance are very strong and you can feel it. You can smell it. But if you start putting the same fragrance, same perfume for a month or for 2 weeks, you don't even feel that smell. Why that happens? Because your receptors adapt to that stimulus. Okay. So based on whether a receptor adapt or not, sensory receptors responses or sensory receptors can be categorized on two types: tonic receptors and phagic receptor. Tonic receptor respond at constant rate as long as a stimulus is applied. So let's see. This is a receptor, resting membrane potential here. And let's see, you give a stimulus like like smell. Smell activates the receptor plasma membrane, and every line is action potential, so they generate. And then you remove it, stimulus, what happens?
Stops creating action potential.
Yeah, so what happens? They still, when you start stimulus withdrawn, there is no stimulus at all. Okay. Now, if you give again another stimulus, again action potential will continue. But in phagic receptor, respond with burst of activity, but quickly reduce firing rate to constant stimulation. And that is called adaptation. For example, smell and touch. So here, you give a stimulus, initially firing rate, you can see frequency is fast, but slowly firing rate decrease. That means these are fast adapting receptors. Everything adapt, but some receptor adapt very slowly, some receptor adapt very fast. And the phagic receptor phase out fast. To remember it, phagic receptor phase out fast, whereas tonic receptor phase out slow. Okay. So pain is tonic receptor and smell and touch, for example, are phagic receptor. Let me give you one example. When you start dating first time and you have a first time touch of each other, you know what is the feeling? What is the intensity of the touch? And after 20 years of marriage, do you think the stimulus will be the same? Nope, it changes. It goes down. People start and leave a good the living condition. There is like everything neat and clean, no bad smell. But let's see if some problem comes, mental problem or other problem, it starts with the same individual. And then they cannot maintain the cleanliness. What happens? They smell, they slowly phase out and they don't see them. Another example, you buy a new bathing soap and you apply first time on your body, you can smell well, but if you use the same soap for a month, you'll not smell the same way. These all are the example of phagic receptors. Okay. Now, the generator or receptor potential. So generator or receptor potentials are the potential in the dendrites because most of the receptors are attached to the dendrites of the nerve fiber. Okay. So here, this is the threshold of the dendritic nerve ending and you guide. So this is the receptor dendrites and 1, 2, 3, 4. So when you give stimulus 1, it is not reaching the threshold. Then you give stronger stimulus 2, stimulus 3, stimulus 4, 2, 3. And when a stimulus 4 reaches the threshold, then what happens? It depolarizes. You know, once you open the threshold, then a lot of sodium channels open, and that gives the initial segment of action. That is the action potential of generator potential because they are generated in the dendrites of the receptors. Okay. So generator or receptor potentials are sensory receptor. They are equivalent to excitatory postsynaptic potential. Do you remember excitatory postsynaptic potential? Excitatory postsynaptic potentials are produced by neurotransmitter binding with the receptors on the postsynaptic membrane. Okay. So these generator potential is produced in response to adequate stimulus. And if threshold reach, generates an action potential, which is number 5. Okay. So how intense that generator potential is based on what is the intensity, stimulus intensity. So generator potential are proportional to a stimulus intensity. So if I pinch someone with less force or a lot of force. If you have a lot of force, then your frequency is high. After threshold is reached, action potential frequency is proportional to amplitude of generator potential. But in phagic receptor, what happens? These generator potential adapt to a constant stimulus. So earlier you had like only fourth level of adequate stimulus, but you need a stronger later. And they quickly diminishes in amplitude. Okay. So here again another way to show generator potential. In tonic receptor, generator potential does not adapt to constant stimulus. You see, action potential. Once you have, they are continuous. Once they are adequate. But in phagic receptor, that change. Okay. Now cutaneous sensation means sensation in the skin. What are the cutaneous sensation in the skin? They are touch, pressure, heat, which is called temperature, cold, and pain. These all receptors are mediated by free and encapsulated nerve endings. I told you earlier, receptors are either free nerve ending, encapsulated nerve ending, modified neurons, or modified epithelial cells. So cutaneous sensation are free nerve ending and encapsulated nerve endings. Free nerve endings mediate heat, cold, and pain. Encapsulated nerve endings mediate sensation to touch, change in texture, and pressure. Change in texture means you touch your table, which is smooth, and you touch the pavement, which is rough texture, or some is smooth, some hard, some soft texture. Okay. For them all, they are separate receptors. So the how this cutaneous sensation takes the sensation from skin to the brain. So let's see the pathway of sensation. Here it is your brain. Here is the spinal cord level. Before I do this, I want you to guys to remember which area of the brain is for primary sensory area, somatic sensory area.
Postcentral gyrus.
Postcentral gyrus. Postcentral gyrus is the part of which lobe of the brain?
The back part. The
Parietal.
posterior.
Back part of the parietal or front part of the parietal?
Back part of the parietal. Posterior to the parietal lobe.
Frontal anterior part of the parietal. Okay. So how this cutaneous sensation takes sense from your periphery to the brain. So here is touch, and then is proprioception, means position of the body. Here is temperature, and here is pain receptor. You can see the path. So as you know, the cutaneous sensation cross the side. So when it comes from right side, it crosses and go to the left side of the brain. And when it comes to the comes from the left side, it crosses and it goes to the right side of the brain. So let's see here. In this case, touch. So touch stimulus through the sensory neuron. It's passing to the posterior column of the spinal cord. And then it passes along, and then when it reaches the medulla oblongata, what happens? It synapses with another neuron and then ascend, cross the side. Decussate, cross. So crossing is in the medulla oblongata. And then it goes through the thalamus and then to the sensory area. So both proprioception and touch cross in the medulla oblongata. Let's see temperature and pain. What are they doing? Do they cross in medulla oblongata or do they cross in spinal cord?
Spinal cord.
Yes, so they cross lower. They cross in a spinal cord. That means if somebody has spinal cord damage in road traffic accident and a spinal cord damage is there, what kind of sensation will be lost easily?
Temperature and pain.
Yep. Okay. So that is the pathway. Cutaneous receptors are here. Free nerve ending, Merkel disc, Ruffini corpuscles, Meissner corpuscles, Pacinian corpuscles. There is structures we talked about. And here is sensation. Free nerve ending is for light touch, hot, cold, and pain. Merkel disc is for sustained touch and pressure. That means deep touch and pressure. Ruffini corpuscles endings are for sustained pressure. Meissner's corpuscles are for change in texture and slow vibrations. Pacinian corpuscles are for deep pressure, fast vibration. So these are the receptors and their sensation associated with them. In the cutaneous sensation, we have pain sensation too, and that is mediated by nociceptors, means pain receptors. These receptors, when stimulate, they release a neurotransmitter that is called substance P or neurotransmitter P. And substance P activate the nerve ending from there. The same receptor, nociceptor, is also activated by the capsaicin. Capsaicin is one kind of chemical compound found in the jalapeno. Jalapeno has that compound, and this capsaicin binds to the capsaicin receptors. And through that, it produces heat. That's why when you eat very hot food, it creates the heat. And if this is a lot, then it also causes pain. Same receptors. Okay. These are the cutaneous receptor, Ruffini's, Merkel disc we talked about. These all are the nerve endings in the skin. You see. Okay. Now, some part of our skin is very sensitive and some part is less. Let's see, if you touch your face and if you touch your back of your thigh or back of your leg, will you feel the same way? No. If you have a cut or pinching in your face and pinching in your calf muscle, will they cause the same level of pain? No. Why? Because this different area has different density of receptors. And based on distribution of the receptors, we can determine the receptive field. Okay, so receptive field in any skin area is area of skin whose stimulation results in change in firing rate of a sensory neuron. Okay, and this receptive field area varies, change inversely with the density of receptor. That means higher the density of receptors, less the receptive field. Higher the density of receptive field, less the receptive field. Lower the density of receptive field, higher the field, receptive field. Okay. So here you go. Back, leg have low density of sensory receptors. Therefore, their receptive fields are large. Fingertips and face have high density of receptor. That's why their receptive fields are small. So let me draw here. So let's see. So let's see, this is the facial area here, and here is your back of the leg area. And each dot is receptors. So if you see the number of receptor in a specific area, and you see so in this area, this much area, and if you see this much area, so receptors are less here. So let's see, if I stimulate in this area, will not feel anything. If I stimulate this area, the receptor, then we feel anything. So the number of receptor per square area of the surface is a lot here. Here is less. So density is high here, density is less here. That's why this area is more sensitive compared to this area. And the distance between, so let's see distance between this adjacent receptor here, adjacent receptor here, and adjacent receptor. Here you can see distance, very low. And these distance are receptive field. Are you following me? So smaller the receptive field, larger the density, more the area sensitive. Smaller the density, larger the receptive field, less the sensitive area. Okay. Okay. So how we measure this receptive field in any body part? We're going to do this lab on Thursday in our lab. Okay. So here, the receptive field is measured by 2-point touch threshold. And 2-point touch threshold is measured by using calipers. You see calipers? So we're going to use these calipers. You're gonna separate apart these calipers and ask an individual to close their eyes, and you touch with both of this point of the calipers and ask, do you feel 1 point or 2 point? As long as they feel 2 point, then reduce the distance between these 2 points and ask again whether the subject feel 2 points or 1 point. Once they start feeling 1 point, that means that is the distance between, average distance between adjacent receptors, and that is the receptive field. Okay, so receptive field is measured by 2-point touch threshold. It is minimum distance at 2 point of touch, which can be perceived as separate. Measure of tactile acuity or distance between receptive fields. So you see here, if you feel 2, that means you are activating 2 sensory neurons. If you feel 1, that means you are activating 1. So immediately before this one, this distance is 2-point touch threshold. So based on the sensitivity of an area in our body, let's see 2-point touch threshold of the body parts. You see? Look at this table, go over it, and then I'm going to ask some to the point testing understanding questions. So this is body region. This is 2-point touch threshold in millimeter. I'm going to pick individuals today. So you have to answer. When I ask somebody's name, that person needs to answer question, okay? Okay, Fatima,
Yes.
tell me which region of the body has the highest 2-point touch threshold?
The upper arm. I mean, sorry, the calf.
The calf. Okay. Thank you. Now, Christina,
Yes.
based on this table, which area has the highest density distribution of receptors?
The calf. You hear me?
Yes, I can hear you. Did you get my question?
Yeah.
Based on this table, which area has highest density of receptor?
The calf.
Density.
The first finger?
The first finger. Yes, it is first finger. That's why their 2-point touch threshold is the least.
My nose.
Because the distance between two receptors are very minimum. Let me ask Bella. Are you there, Bella?
Yes.
Based on this table, which area has the least sensitivity to touch?
The calf?
This shows you guys did not get it. Did I say least or most?
Oh,
I think, no, I'm sorry, you are right, you're right. I said least or most.
You said least.
Yeah, if I said least, that's why this is right then. So calf is the least sensitive. Based on this table, somebody else.
Are you looking for someone else to answer?
Yes.
Okay.
Gurbir,
Yes.
based on this table, which area has the least distance between two adjacent receptors?
The big toe,
Why?
because it's the furthest.
I think it's the first finger.
Is it the first finger?
Yeah.
Yes, it is the first finger. That's why their
There's
two
very only
point
2 points.
threshold is the only 2. Okay, so this is the 2-point touch threshold. You don't need to memorize these things, but you need to know the concepts. Okay. So we are done the first part, and I'm gonna bring the another slide. Stop share this one. Did we not finish last time the central nervous system?
We didn't finish
With-
the autonomic nervous system.
Okay, I said I have no time to finish autonomic nervous system in the, let me stop first the recording. Why we put these things in recording?